Right now, inside your pinky finger, there is a sprawling microscopic metropolis.
Oh, absolutely. Millions of packages are just, being shipped across these tiny microscopic highways.
Yeah, and you've got specialized power plants burning fuel, massive command centers sending out, like, encrypted codes.
Heavily guarded borders, constantly monitoring traffic. It's bustling, it's chaotic.
And it is happening every single second. And the wildest part, we are just talking about one single living cell.
Right. It is a scale of activity that is, honestly, it's almost hard to comprehend.
I mean, we walk around completely unaware of the trillions of these microscopic cities functioning inside us. And, inside every living thing around us, too.
Which is exactly why today's deep dive is going to completely reframe how we look at the natural world.
Definitely, because this deep dive is actually specially tailored for those of you diving into this FPS and catchiest science curriculum.
Yeah, just like the pilot lessons that students are experiencing right now in the Kodiak and Kuskook school district.
Exactly. So whether you are observing the kelp washing up on the shores of Kodiak, or examining spruce needles in Kuskook, or, honestly, just looking at the skin on the back of your own hand.
You are looking at incredibly complex infrastructure.
But so often in school, a cell just gets described as a bag of parts.
Right, the classic bag of parts.
Yeah. We get this image of a tiny water balloon just filled with floating vocabulary words.
And that bag of soup model completely misses the reality of biology.
It really does.
Yeah.
Because our mission today is to show you that a cell is the smallest unit of matter defined as alive, but it only achieves that life because it operates as a system.
Yes, a highly coordinated dynamic system. The word system is like the key to everything we're going to talk about today.
Because every single structure inside a cell has a specific job, right?
Yeah.
But none of them can work in isolation.
Exactly. It is an integrated physical network.
So before we can explore the inner workings of this microscopic metropolis, we have to figure out how to actually get inside.
Right. Every city has a border.
Yeah, so how does a cell protect itself?
Well, a cell operates as an open system bounded by a flexible, selectively permeable membrane.
Selectively permeable, that's a big one.
It is. It's just the scientific way of saying it doesn't let just anything in or out. It acts as like strict border control.
Okay, so it's deciding what crosses.
Right. It brings in vital inputs like water, oxygen, and glucose, which is a simple sugar. And it ensures that dangerous outputs like toxic cellular waste or excess carbon dioxide are flushed out.
So it's kind of like the world's most sophisticated laboratory bouncer.
Yeah.
Yeah. Like checking ID badges at the door.
Well,
making decisions about who gets into the club and who gets kicked out.
it is tempting to think of it that way, but we have to be really careful with that metaphor.
Well, why is that?
Because a cell membrane doesn't have a brain, it isn't making a conscious choice.
Oh, right. Okay.
A better way to visualize it is like a highly specific microscopic coin sorting machine.
A coin sort.
Yeah, so the membrane is embedded with these physical and chemical channels. If a glucose molecule bumps into the membrane, and it has the exact right physical shape and the exact right magnetic charge.
It just drops in.
Exactly. It slides perfectly through the slot and enters the cell.
But if a toxic molecule bumps into that same slot...
The shape is wrong, the charge is wrong, and it just physically bounces off.
Oh, wow. So it's purely mechanical and chemical.
Completely.
It's just a perfectly designed lock and key system happening millions of times a second.
Right. And by constantly filtering these molecules through those slots, the cell membrane maintains homeostasis.
Homeostasis, another huge vocabulary word for the listeners.
Yes, and a crucial one. Homeostasis is the ability of an organism to maintain internal balance, despite, chaotic external pressures.
So if the environment outside the cell suddenly gets too salty or too hot,
the membrane's channels react to keep the inside chemistry perfectly balanced.
wait, what happens if it can't? Like, what if the environment is just too extreme and homeostasis completely fails?
Well, the cell doesn't just quietly fade away.
It doesn't.
No, often, if a cell realizes its internal balance is irreparably broken, it activates a self-destruct sequence.
Wait, seriously?
Yeah, it deliberately dismantles itself so that its failure doesn't harm the rest of the organism. It's a remarkable built-in failsafe.
That is intense. Now, I have looked through microscopes in science class before, and I remember looking at plant cells, specifically from an aquatic plant called Elodia.
Ah, yes, classic classroom example.
Right, and they did not look like flexible, squishy membranes. They looked like rigid, bright green bricks just stacked on top of each other.
They do look like bricks, yeah.
So why are plant cells so much stiffer than animal cells?
Well, that difference comes down to the environment the organism has to survive in. If you look at human cheek cells, for example,
Which are animal cells.
right, they are transparent and flexible because they only have that cell membrane. Humans have a skeleton to hold us upright.
Yeah, we have bones.
But a plant doesn't have bones. It has to grow upright to reach the sunlight using only its cells.
Oh, I see.
So outside of their flexible cell membrane, plant cells build an additional boundary, a thick, rigid, cellulose-based cell wall.
And that is what gives them that box-like shape.
Exactly.
Wait, I have to push back on this a little bit.
Okay, go for it.
If a plant cell is locked inside a rigid, impenetrable brick wall, how does the coin-sorting membrane get anything?
That is a very logical question.
Right, like how does water or glucose get to the slots if they are trapped behind a solid wall?
Well, the cell wall is rigid, sure, but it is not a solid brick wall. It is actually highly porous.
Oh, so it has holes in it.
Lots of them. Think of the cell wall more like a sturdy chain-link fence surrounding a building.
Okay, I can picture that.
The fence provides structural integrity and keeps large threats out, but rain, air, and small materials can easily flow right through the gaps in the chain link.
Oh. And once those inputs pass through the plant cell wall, they hit the flexible cell membrane just beneath it.
Exactly, where the coin sorting channels do the actual filtering.
So the fence gives the shape, but the coin sorter still controls the traffic.
You've got it.
That makes perfect sense. Okay, so we've made it past the chain link fence, we've slid through the chemical slots in the membrane, and we are finally inside the cell.
Welcome to the Bioforge.
Right. What is actually running this city?
Inside, you find the organelles.
Organelles, so like tiny organs.
Yeah, essentially specialized subcellular structures. You can think of them as the specialized departments of a factory.
Okay.
Some handle the blueprint, some handle the energy, some handle the waste.
And what's the most important one?
The most prominent is the command center, the nucleus. It serves as the information storage subsystem.
The storage subsystem, meaning it holds the DNA.
Exactly. It houses the DNA, which are the literal blueprints, and it directs other organelles to synthesize the specific proteins the cell needs to repair itself and function.
But wait, if the nucleus is constantly sending out blueprints and ordering the construction of proteins, that has to require a massive amount of energy.
Oh, a ton of energy.
A command center needs a power grid. So where is the cell getting the juice to do all this heavy lifting?
That brings us to the power plants of the system, the mitochondria.
A powerhouse of the cell.
We hear that phrase a lot, but let's break down what it actually means. Remember those inputs the membrane allowed in?
Yes, specifically the glucose and the oxygen.
Right. The mitochondria take those raw inputs and perform cellular respiration.
Now, I've heard that term, cellular respiration, but what is actually physically happening inside the mitochondria to make power?
It all comes down to breaking chemical bonds.
Okay, breaking bonds.
Yeah, a glucose molecule holds a lot of potential energy locked in the bonds between its atoms.
Like a coiled spring.
Exactly like a coiled spring. The mitochondria use oxygen to physically break those bonds apart, and when those chemical bonds snap, they release energy.
Oh, wow.
The mitochondria capture that released energy and package it into a highly usable molecule called ATP.
So glucose is the raw fuel,
Hmm.
but ATP is the actual electricity that powers the machines. It's like taking a gold bar to the bank. A gold bar is valuable, but you can't, use it to buy a sandwich.
That is a highly effective way to visualize it.
So the mitochondria exchange the glucose gold bar for ATP, which is the actual cash the cell uses to get work done.
Right. Every single organelle relies on that ATP cache to function.
Got it.
Now, going back to those green plant cells you saw under the microscope.
The Elodia cells.
Yes. Plant cells have an entirely different specialized energy organelle that animal cells simply don't have. Chloroplasts.
Oh, right, because plants don't eat food. They have to make it.
Exactly. Chloroplasts essentially act as microscopic solar panels. They capture light energy from the sun and use it to literally manufacture their own glucose from scratch.
Which they then send to their own mitochondria to turn into ATP.
You've got the whole system down.
This all loops perfectly back to the idea of the cell as a system. The connections are totally dependent on each other.
Completely dependent.
Think about it. If the membrane's channels fail and stop letting oxygen in, the mitochondria have nothing to mix with the glucose.
And if they can't break those bonds, they stop producing ATP.
Right, and if the ATP power grid goes down, The nucleus loses power and can't send instructions to build proteins.
And without new proteins, the cell membrane can't repair its chemical channels.
If one part drops out, the entire system crashes.
You cannot separate the function of the membrane from the function of the mitochondria or the nucleus. They are a single, continuous, interacting network.
Which is mind-blowing when you remember that this entire complex network is completely invisible to the naked eye.
It really is.
Which brings up a historical mystery I just don't understand.
Oh, what's that?
Well, if we can't see this stuff, how did early scientists ever figure out these specific mechanisms existed?
That is a remarkable story of how science is completely dependent on its tools.
Okay, lay it on me.
So in the 1600s, an English scientist named Robert Hooke looked at a piece of cork under a very primitive microscope.
Cork, like from a tree. So plant cells.
Exactly. And because plant cells have those thick, rigid cell walls we discussed, the empty structural boxes were incredibly obvious to him.
The chain link fences.
Right. He called them cells because they look like the small, square rooms that monks lived in.
Okay, so he saw the chain link fences. But what about the membrane? When did they find the coin sorter?
Believe it or not, the flexible, transparent cell membrane went completely undiscovered for another 200 years.
Wait, 200 years?
Yes. The progression of cell theory totally stalled out.
But why did it take them so long to find the most important security border of the cell?
Because of severe defects in glass manufacturing.
Seriously, the glass was just bad.
Yeah. The lenses in early microscopes had terrible chromatic aberration. When you tried to magnify something as incredibly thin and transparent as a cell membrane, the glass would bend the light incorrectly.
And it just looked blurry.
The image just turned into a blurry, colorful mess. Science was trapped by the limits of human engineering.
Wow. So when did it change?
It wasn't until the late 1800s that a physicist named Ernst Abbe figured out the complex mathematical formulas required to manufacture glass lenses without those distortions.
So better math led to better glass, which led to better biology.
Precisely.
But even with clearer glass, a membrane is mostly transparent. How did they definitively prove it was there, holding the inside of the cell together?
They proved it through the mechanism of osmosis.
Osmosis?
Yeah, two scientists, Nageli and Kramer, were observing plant cells. They decided to flood the outside environment of the plant cell with a heavy saltwater solution.
Now, osmosis is about water moving to balance things out, right?
Correct. So when the environment outside the cell suddenly became incredibly salty, the water inside the cell rushed out to try and dilute that salt.
Trying to maintain homeostasis.
Yes.
Yeah.
And as the water drained out of the cell, Nageli and Kramer watched through their newly improved microscopes as the green inner contents of the cell shriveled up.
Whoa.
And it actually pulled away from the rigid outer cell wall.
Oh, because the inner bag was shrinking.
Yes. By watching the inside physically detach and shrink away from the rigid wall, they proved there had to be an invisible, flexible boundary holding the internal organelles together.
That is brilliant. They couldn't see the membrane itself, but by watching how the system reacted to salt, they deduced it had to be there.
Exactly.
Which highlights such a crucial point about how we learn. We tend to think of science as just a textbook of undisputed facts.
Right, a static list of things to memorize.
Yeah, but it's actually an active, ongoing struggle to build explanations using whatever tools we have at the moment. When we can't observe something perfectly, we have to build models.
And models are an active process of explanation building. A model isn't just a random guess, you know?
Right.
Every single part of a conceptual model must be justified by real-world data. And if the output of a model doesn't match the real world, scientists have to revise or scrap it.
Do you have an example of that?
Sure. For a long time, early scientists had a model that allowed for spontaneous generation. Spontaneous generation. That's the idea that life could just magically emerge from non-living matter, right?
Right, like thinking maggots just magically appeared out of old meat because they didn't have the tools to see the microscopic eggs.
Because they couldn't see them, their model just said, well, they just popped into existence.
Exactly. But eventually, other scientists gathered new data proving that cells only ever come from pre-existing cells.
And the old model couldn't explain the new data?
Right, so the spontaneous generation model was thrown out. Science isn't about protecting the right answer. It's about constantly exposing our own misunderstandings and refining our explanations.
And that idea changes everything about how we should approach learning today.
It really does.
Because science isn't about sitting in a classroom trying not to get the wrong answer on a multiple-choice test. It's about taking your current misconceptions and working through them.
Right, I mean, if you've never seen a cell, It makes perfect logical sense to think of it as a bag of soup. Exactly. A misconception is just a logical attempt to make sense of your experiences until you get better data.
And this is why stepping out of the textbook and observing the natural world is so important.
Definitely.
When we talk about living systems, the same principles apply whether you are looking at a microscopic single cell or an entire sprawling ecosystem.
And for our listeners in Alaska, you have access to some of the most complex, observable living systems on Earth.
You really do.
You can walk outside and look at the fireweed growing, or the moss clinging to a rock, or even the microscopic phytoplankton blooming in the bay.
Yeah, and every single living thing in those environments is operating using the exact same cellular systems we are talking about today.
Right. The chloroplasts in the kelp are capturing the sunlight, the mitochondria in the brown bears are snapping glucose bonds to generate the massive ATP needed to hunt salmon in a rushing river.
The microscopic powers the macroscopic.
And this highlights the incredible value of local knowledge and traditional practices, too.
Absolutely. For generations, people have carefully observed how these natural systems interact.
It's like a traditional fish trap, for instance.
Right. A traditional fish trap works because it is designed with a deep observational understanding of the river's natural flow and the behavioral systems of the fish.
And when you combine those deep traditional observational routes with scientific tools.
Like microscopes and biological models.
Exactly. They work hand in hand. They both deepen our understanding of how living systems adapt, maintain homeostasis, and survive.
It's all connected.
Okay, let's bring this all together. We started this deep dive by shrinking down and stepping into the metropolis, the cell.
And we learned that it is definitely not a random bag of parts. It is a dynamic, tightly integrated system.
Right. We explored how the rigid cell walls provide structure, while the flexible cell membranes act as specific chemical coin sorters to maintain homeostasis.
We saw how the command centers in the nucleus rely entirely on the energy produced by snapping chemical bonds inside the mitochondria and chloroplasts.
Because every individual structure is completely dependent on the whole.
Exactly.
And we learned that the scientific tools you use and the observations you make in your own local environment are just as vital today as they were to the scientists who first looked through those distorted glass lenses.
Now keep questioning the systems around you and recognize that the environment you observe every day is built from these incredible microscopic interactions.
We want to leave you with a final thought to mull over, something to discuss in class, or just think about the next time you see a brown bear, a piece of fireweed, or even just look at your own hand.
Here is your question.
How does understanding the parts of a cell help us understand the whole living organism?